What if humanity gained the power to rewrite its own genetic code and then use that power before fully understanding the consequences? For centuries, diseases caused by faulty genes were considered unavoidable. But in the 21st century, scientists developed a revolutionary technology that promised to change that reality forever. Its name was CRISPR. Often described as genetic scissors, CRISPR-Cas9 gave researchers the ability to edit DNA with remarkable precision. For the first time in history, scientists could potentially remove harmful mutations and prevent certain genetic diseases before they even developed. The technology was hailed as one of the greatest scientific breakthroughs of modern medicine. Researchers envisioned a future where diseases like sickle cell anemia, cystic fibrosis, and muscular dystrophy could be cured at their genetic roots. But while the possibilities were extraordinary, so were the risks. Many experts warned that editing the DNA of adult patients was very different from editing human embryos. Changes made to embryos could be inherited by future generations, meaning any mistake could permanently alter the human gene pool. For years, scientists urged caution. Then in November 2018, the world received shocking news. A Chinese scientist named He Jiankui announced that he had successfully edited the genes of twin girls before they were born. Using CRISPR technology, he altered a gene called CCR5, which plays a role in HIV infection. His goal was to make the children resistant to the virus. The announcement stunned the scientific community. For the first time in history, genetically edited babies had reportedly entered the world. What made the experiment even more controversial was that the children were not suffering from a genetic disease that required treatment. Existing medical methods already existed to reduce the risk of HIV transmission. Many scientists immediately questioned whether the procedure was necessary at all. The concerns extended far beyond the experiment itself. Although CRISPR is highly precise, it is not perfect. Researchers knew that unintended genetic modifications known as off-target effects could occur. In simple terms, the technology could accidentally edit parts of the genome that were never meant to be changed. Such unintended alterations might increase the risk of cancer, developmental disorders, immune system abnormalities, or other health complications that may not become apparent for years. The greatest concern was that these changes might not stop with the children. Because the editing occurred at the embryo stage, future generations could inherit the modified DNA. In other words, a mistake made in a laboratory could potentially affect descendants who had not even been born. The backlash was immediate. Scientists, ethicists, governments, and regulatory agencies around the world condemned the experiment. Many described it as reckless, unethical, and scientifically premature. The controversy quickly evolved into a global debate about the future of genetic engineering. Should scientists be allowed to edit human embryos? Who decides which genetic changes are acceptable? Where should society draw the line between curing disease and enhancing human traits? Could gene editing eventually lead to so-called designer babies, where characteristics such as intelligence, appearance, or physical abilities are selected before birth? These questions suddenly became real. Investigations later revealed that the experiment violated ethical guidelines and regulatory requirements. Chinese authorities determined that proper oversight had not been followed and that scientific standards had been ignored. The researcher was eventually convicted and sentenced to prison. But the impact of the experiment extended far beyond one individual. It became a turning point in the history of biotechnology. Governments strengthened regulations surrounding human gene editing research. Scientific organizations introduced stricter ethical guidelines. International discussions intensified regarding the responsible use of CRISPR technology. At the same time, researchers continued exploring the enormous medical potential of gene editing. Today, CRISPR-based therapies are helping treat diseases such as sickle cell disease and beta thalassemia, offering hope to patients around the world. The lesson from the 2018 experiment remains clear. Scientific progress must be guided by ethics, responsibility, and caution. The wrong CRISPR experiment showed the world that while humanity now has the power to rewrite DNA, it must carefully consider how that power is used. Because when you edit the human genome, you are not simply changing a single life. You may be changing the future of humanity itself.